Right, I am delighted and honoured to welcome Professor Benton to speak to us today. It's very kind of you to make the time for us. And given your background, I am doubly honoured. Professor Benton is the Professor of Vertebrate Paleontology at the School of Earth Sciences at Bristol University. He's fascinated by the transformation of paleobiology into a testable science, and one of those discoveries determining the colour of dinosaurs was rated as one of the top scientific discoveries of the 2010s. He works on large-scale evolution of major groups such as dinosaurs, exploring the effects of mass extinctions, environmental change, and biological innovation on the evolution of reptiles, birds, and mammals. And he works with fossils to build evolutionary trees and uses them to date major events and rates and patterns of evolution to help us understand why some groups of animals are more successful than others. I mentioned his teaching earlier. To date, and these numbers may have been, surpassed since then, I do admit, you've supervised more than 70 PhD students, founded the Bristol MSc in Paleobiology, which has welcomed over 400 students so far. And you have over 400 scientific papers and more than 50 books to your name, which is impressive by anybody's standards. And I'm also pleased to say that you have been honoured. In 2010, you had a rhynchosaur named after you, Bentonix, which means Benton's claw. And in 2014, you were made a Fellow of the Royal Society for your fundamental contributions to understanding the history of life and particularly biodiversity fluctuations through time. So if I've missed anything, please do tell. And on that, I will hand over to you.
Thank you very much indeed, Mary. And I will just share the screen and make a start. Here we go. Right. Yes, that's it. Okay, so thank you all. And I'm going to talk about a book we wrote, but also explain something about how we can study dinosaurs, in fact, study any ancient times, and consider the sorts of evidence that we use. And color is a great topic because, of course, it's one of those things which people would surely have said, we'll never know. And yet we actually do. So let me tell the story and see how we get on. Right, there we go. So my introduction, yes, as Mary kindly said, I've written lots of stuff and various books. And in fact, the talk I'm giving relates to a particular book as well, with wonderful illustrations by a very excellent paleo artist, which I'll talk about. But what I want to do is to go back 30 years to the year 1996 and tell a story about when we didn't know anything about dinosaurs with feathers and how that knowledge has really boomed in the last 30 years. So I went as a much younger person to the annual meeting of the Society of Vertebrate Paleontology in New York that year at the American Museum of Natural History. Here it is, looking over Central Park. And this is a big meeting with 1,000 or more people attending. And during the meeting, halfway through, this article appeared in the New York Times, written by Malcolm Brown, who was one of the very respected science writers for the New York Times, and talking about a new fossil dinosaur with traces of feathers. And this was a very new idea. And the headline, with sufficient caution, but nonetheless boldness, feathery fossil hints, dinosaur bird link. And the name Philip Currie is mentioned there at the bottom. I'll just see if I can do this. There we go. So Philip Currie is a name I'll mention as well. He's a very noted dinosaur expert. And what had happened was at the meeting, and I saw this, there was a first delegation of Chinese scientists attending. And this was a great novelty at that time because China had been a very closed country. And Peiji Chen from the Nanjing Institute of Paleontology had brought a photograph of an unusual specimen, this one here, and he was showing it. And this thing is about a meter long, and in the photograph in color, you could see the skeleton with the head over on the right and the back, and then a very long tail and the arms and the legs. And it's obviously not a bird. The arms are very short, although it has a somewhat bird-like head. But what he was drawing attention to were this fuzz along the back of the head, the back, and then on the tail. And it appears to be somewhat tufty in the tail region. And then within the rib cage was an area of black and brown organic matter. And nonetheless, he was showing it to all the dinosaur experts of the day and asking, well, what did they think? And they could identify that it belonged to a group that was already known, the Compsognathids, known from Germany. And yet this thing appears to have feathers. And yet one person at the meeting in New York knew more, and this was Phil Curry, because he was aware he had been in China just the week before, and he had seen this specimen, which shows the same features, but it's kind of looking the other way. And this was in the course of being described already by scientists in Beijing. And they were at the National Geological Museum of China, and they published this and named it as Sinosauropteryx prima in a paper that was published about the same week as the New York meeting in the Chinese language, though. So it was only translated into English later. And you can see it's a short paper, 3 or 4 pages. So they were determined that this was a bird, aves. So they're calling it a bird because it's got feathers. Peiji Chen and his colleagues from Nanjing eventually published their paper in English in Nature 2 years later. And so this is their specimen, this is their paper. And of course, this paper attracted a lot more attention because it was in English, it had color illustrations and was very prominent. It was in the journal Nature. But what was going on? Because here is the holotype specimen of Sinosauropteryx Museum in Beijing. Here is the Nanjing specimen, and evidently they are one and the same. They are counterparts or opposite slabs of one single specimen. I don't know if they've ever been in the same room together since 1996. They clearly were when they were discovered, and then some collector farmer had a nifty idea. Let's double my money by selling it to 2 different institutions. And that's what happened. Nonetheless, this is important because, of course, it was the first dinosaur with feathers. And it was helpful in a way that it belongs to a known group, so we know where it sits on the evolutionary tree. And let's just put this into context because when this was discovered, our knowledge of the evolution of birds was quite limited. Of course, we have Archaeopteryx from the Upper Jurassic 150 million years ago. And famously, Huxley jumped at this as a perfect proof of evolution. He was Darwin's great friend. And only a year or two after Darwin's Origin of Species was published, here was the perfect intermediary fossil, half reptile, half bird. And Thomas Huxley very famously said that Archaeopteryx was a dinosaur in birds. And so there were lots of shenanigans and to and fro's, of course, over the years. But broadly speaking, this is the picture that we had in 1990. We have Archaeopteryx as accepted by everybody as the first bird at 150 million years ago. We have, of course, the huge diversity of modern birds. There are maybe 10,000 species of birds, and they have a, a reasonable fossil record back to about 75 million years ago. In the late Cretaceous, we know of some early duck fossils and other groups at that time. And then birds diversified enormously in the Cenozoic after 66 million years ago. But there was a long gap in time without very many helpful fossils, all the way then in the other direction back to dinosaurs. And so this was unfortunate because, of course, in all the textbooks of evolution, Archaeopteryx was the classic missing link, the indicator of evolution in action. And of course, the origin of birds is just one of those great stories of evolution that that people can really understand, and it really shows the wonderful nature of evolution, the innovation of feathers and flight, how important that was, and the fact that we can try to interpret some aspects of how that happened using the fossils. But we didn't have very many. Then we moved to China. This is where Sinosauropteryx came from. This is Sihetun Quarry in Liaoning province in the northeast of China. So here is Beijing with the red star, and this is Liaoning. So we drove in my first visit to China about 15, 20 years ago. We drove from Beijing to Liaoning, which doesn't look very far, but of course, remember, China's awfully big. So this is 500 miles, that distance across one province. And this one quarry in Liaoning produced 1,000 specimens of Confucius ornis. And at the time these were being discovered, Archaeopteryx was only known from about 10 or 12 specimens. And each of those was guarded like the crown jewels. And so these localities in China were not only producing birds, but all sorts of other fossils, including dinosaurs. And all the indications are that these are lake deposits with aquatic arthropods and fishes and planktonic creatures and plants that would be living in the water, as well as occasional swimmers like turtles and dinosaurs and mammals and things that would be flying over the pond, like pterosaurs and birds.
Right.
And they all seem to preserve soft tissues. So here is a skeleton of a bird, and there's some traces of organic matter and the stomach contents. Its stomach area is full of seeds, and they can be identified as to which plant the thing was eating. So here's Sinosauropteryx. Here's the specimen. It was the first feathered dinosaur to be reported. These are bristles that can be seen all the way down the back, and it seems to be closely related to Compsognathus from the same localities as Archaeopteryx. But what are these feathers? Are they really feathers? Of course, at the time, this was heavily debated. And here is an illustration from the 1998 paper. There were some additional specimens by then. And in close-up, you can see the bones are a sort of pinkish color. And these strands of dark brown are what were being called proto-feathers. Some people said they were shredded skin, but that's quite a hard argument to maintain because we don't know any process whereby skin would shred in such an equable manner. And when they were analyzed chemically and in detail, they showed all the characteristics of hairs and feathers. Further specimens of different species were found. This is a different kind of dinosaur belonging to a group of oviraptorosaurs. And in addition to the downy, whiskery little feathers, it's also got pennaceous feathers on the wings. So these more complex feathers with a central quill and lateral barbs. And even though we could call it a wing, it's clearly not big enough for flight. And then there's a nice tuft of these pennaceous feathers on the tail. A very weird looking creature. And at the same time, yet another dinosaur was noted. Here's Microraptor. This was named. And the specimens are amazing. You can see the whole skeleton with the wings and the hind limbs. And here's the head and the long bony tail. And like Archaeopteryx and the others, it's got long flight-like feathers, pennaceous feathers along each of the wings. But blow me, it's got them on the hind limbs as well. So this is a tetraptor or quadriraptor. Part of its flight area, the flight surface, is on the hind limbs. And indeed, this is looking more as if it might be capable of flight when you look at that area of feathered wing that's available to it. Obviously, the birds were abundant as well. So Confucius ornis shown here, this amazing specimen. People used to rave about Archaeopteryx, and they still do. But nobody's yet found a single slab with two specimens on it, let's say, like this one. And not only two random specimens, it appears to be a male and a female. The long-tailed male with these sort of display feathers and the short-tailed female. And you can see that they are similar to the dinosaur. They are surrounded by the same fuzz of feathers that the dinosaurs show as well. And these are birds anatomically. There is no question. They've got the reduced bones of the forelimb and other features in the skull and the pelvis. So numerous wonderful and amazing fossils that were just booming out of China and really revolutionizing our view. And why does all of this matter? They're not just curiosities or oddities. Remember that figure I showed a moment ago of our inadequacy of knowledge in 1990? By the year 2000, by the year 2010, we had been able to, we, the paleontologists, had been able to fill in an enormous amount of detail, both below Archaeopteryx and above Archaeopteryx in the evolutionary tree. So it was filling up the detail. And then you can look at individual structures like the forelimb shown across the top here, the evolution of a wing. I think this is a lovely artwork from National Geographic showing Sinosauropteryx over on the left with no pennaceous feathers, and then various dinosaurs like Caudipteryx with a limited amount. Here's Archaeopteryx in the middle, clearly capable of flight, and then a number of fossil birds, and then modern birds over here. So part of the importance of finding these new fossils is, of course, it helps us to fill out our knowledge. And for anybody who asks questions, how do we know about the evolution of birds? We can give a much richer answer than we would have been able to do a few years ago. So that's great. And that allows us to do further analysis. So here in more detail, this is essentially the same diagram, but in much more detail against the geological timescale. So the counting back from the present day, the Cretaceous Jurassic boundary is at about 145, somewhere about here. So this is Archaeopteryx just below the line, and a lot of these Chinese things are coming from 120 to 100 million years ago. So this is a very detailed evolutionary tree of the theropod dinosaurs through to birds. And this highlights the fact that whereas some theropods get to be big, this is T. rex right up here at the end of the Cretaceous, and there were a number of other giant theropods, and everybody loves them. They could eat anything, and they're huge and terrifying. However, along another major branch of evolution, they were getting smaller. And of course, there are two ways mechanically to become capable of flight. One, of course, is for you to stay the same size, but to grow enormous wings that would enable you to fly like an angel or something of the kind. The other solution, maybe easier, is just to get very small. And that's what was going on, that during the Jurassic, with this particular branch of theropods, the Maniraptorans, they were not only getting smaller, but their wings were sprouting. So remember one thing about the giants like T. rex, their arms were getting shorter, and to the point that they were pretty much useless, it seems. They were feeding by using their feet to hold down the prey and just biting at the prey with their mouths. The hands were not really much used. Whereas on the other hand, the Maniraptora, as the name suggests, they were the hand hunters. Their arms and hands were getting bigger. They were getting feathered. They were acquiring pennaceous feathers. And by getting smaller and smaller, they got to the point of Archaeopteryx weighing less than a kilogram, at which point the wing area was big enough that it could take off. And not only were birds and their ancestors mechanically capable of flight, a whole bunch of other early Maniraptorans too, including Microraptor, which we saw earlier, which I think is right up, is somewhere up here. Where's Microraptor? There it is. And a number of other groups. So here's Microraptor over here with its four wings. There's this weird dinosaur with bat-like wings and feathers. And if you ever want to know the shortest dinosaur name ever, this is it. Ichi is the shortest name, and I don't think you could get shorter than that for the genus and the species name. And so this is a new discovery. Not only did we learn that flight was due to miniaturization and that took maybe 50 million years, as we saw in the previous slide, but also, excuse me, that flight originated independently multiple times. And we kind of can see that because the strategy of Archaeopteryx is the same as in modern birds, two forewings. The strategy of Microraptor, which is sort of related, but a bit distantly, it's a different strategy. It's got four wings. And the strategy of Ichi, again, is different. It's got bat-like membranous wings. So That's two things, I guess, we've discovered. We filled out the knowledge of the fossil record so that we can much better explain the timing and the process, the anatomy of how flight originated. Secondly, we discovered unexpectedly that lots of these early dinosaurs actually, to some extent or another, were capable of flight, whether gliding flight or flapping flight. And thirdly, the feathers enabled us to discover color and to reconstruct some of these dinosaurs in color. So here we are. This is our paper from 2010. It's in the journal Nature, so it's prominent. It took a lot of effort to convince the reviewers, but we eventually got through. And you can see it's a smattering of Chinese and British authors. So this was based in work done from Beijing and Bristol. And we are making a definite statement. And the one word I want you to notice here is melanosome. I'll be coming back to that. This is the key. It obviously raised a huge amount of publicity at the time because, of course, to claim that you've got the color of a dinosaur is a pretty daring claim. And it was not only our work, we were studying Sinosauropteryx, this is our reconstruction, but a rival team at Yale University and with Chinese colleagues was studying a different dinosaur, Anchiornis, and they used the similar method and came up with a very detailed reconstruction of the colors and patterns in that dinosaur. And to make it really clear, we are definitely saying this is not based on an opinion, but this is based on evidence. So we're saying we can now look at paintings, reconstructions, and we can say, for example, this one on the left is incorrect, and this one on the right is more or less correct. And we can point to the evidence in different parts of the body, in the tail, on the back, in the head crest, and later work around the face and the belly. So we've sampled from all over. So how do we do it? It's based on these structures called melanosomes, which are the pigment-bearing capsules within the feathers. So the melanin, of course, let's start with melanin. It's a very well-known pigment. Most people have heard of melanin, and we associate it with dark colors. And the common type of melanin in our skin and hair and indeed in all organisms. Melanin occurs very widely in animals and plants. It's in our internal organs. It's everywhere. It's within the liver, the spleen, the brain, as well as providing color in the skin and the hair. Um, and when color enters hair or feathers, hair and feathers are made of the protein keratin, which is the same as the protein making your fingernails. So the natural color of keratin is transparent, although we call it white when we see it expressed. But if you look at your fingernails, they are the color of the underlying skin, and they do not carry any pigment. Whereas hair and feathers do. And the way the pigment enters the hair or the feather is through the follicle as the hair or feather is emerging in the skin. The skin cells can make two kinds of melanin, eumelanin, which gives black, brown, and gray colors, and pheomelanin, which can give ginger colors. And the two kinds of melanosomes, these different chemical forms of melanin are enclosed in, they differ. So eumelanin are in eumelanosomes, which are sausage-shaped, whereas pheomelanin, giving a ginger color, are in spherical pheomelanosomes. So this ginger patch on the zebrafinch is due to close packing of pheomelanosomes full of the gingery pheomelanin. Whereas all the other colors on the zebrafinch, the black and the dark brown and the gray, and even the blondie color of the belly, they come from eumelanosomes. And the intensity of the color depends on the packing, how much. And a single feather like this one here at the bottom can show a variety of colors. And it's extraordinary how the feathers of a bird can have these different colors and yet put them all together. And they provide a very exact patterning. So I think this feather is from the wing cover area. So you've got two white spots and a gingery color, and it's sort of darker deep underneath. So we took samples of the feather-like structures from the skeleton of Sinosauropteryx, looked at them under the scanning electron microscope, and this is what we saw. And we compared, so we looked at this. They're moldic, which means mostly they are hollows in the modified keratin of the feather. Here are one or two whole melanosomes, but they're mainly moldic because, of course, when the specimen was split open, the bird fossil, the dinosaur fossil has been sitting flat in the rock. And when the two pieces of rock are broken open, like opening the pages of a book, a lot of dust and stuff falls out, and that was swept away by the collectors, leaving a kind of moldic appearance. So let's refer back to our current day evidence. Which one does it look like most? It looks most like this one at the top, pheomelanosomes giving ginger color. And so this is our inference. And what we call this then is a chain of inference, which is testable and it can be refuted. So any critic, anybody who, and this is the nature of science, of course. Science it consists of working hypotheses, which are the best explanations of the data. And so the best explanation of this data is that Sinosauropteryx was colored ginger. And it had a ginger and white stripy tail. It had more solid ginger over the back of the head and the neck. It had a counter-shaded pale colored belly and underside. And in fact, also had a kind of bandit mask, which we'll see in a moment. So how could you disprove it without a time machine? You don't need a time machine because you first of all look at the fossil, and the critic would say, we need some repeatability here. Let's look at a different fossil. So you get another specimen of Sinosauropteryx, and you persuade or attempt to persuade the curator to let you loose on it with your scalpel. Secondly, the critic might say, oh, well, this is some nonsense that the Bristol team have worked out with their SEM. This is an artifact. It's not real. It's some sort of thing they made. They've cheated, or they've not cheated, but they've been deluded. So, of course, the answer to that is obvious: repeatability. The critical team is perfectly at liberty to investigate the specimens on their own scanning electron microscope, or indeed any other appropriate instrument to see what they can find. And then thirdly, we could criticize the uniformitarian evidence. If it is found that this relationship of shape and chemistry is not constant and does not map in a direct way from eumelanosome through eumelanin to ginger color, and likewise for the eumelanin to the darker colors. If exceptions are found, then we'd have to look again. And I should say these relationships here between shape and color apply also in mammals, including humans. So it seems to be a very constant feature of vertebrates at large. It's not just a feature of birds, because the fact that it's vertebrates at large gives us some confidence, perhaps, that we can apply it to fossils. So we would say this chain of inference is scientific, it's testable. You don't need a time machine. We know the colors and the patterns of Sinosauropteryx. What becomes speculative, of course, is what's the function? Why would it have those patterns? And you could evaluate different ones. You could say maybe camouflage. Think about zebras and tigers. They have stripy tails, but also they have stripy bodies too. So the only camouflage aspect probably is the counter shading, which of course a lot of mammals have. And that is a well-known effect of rendering a three-dimensional animal more or less two-dimensional. So that's very likely camouflage. But the stripy tail, and if we go back to Anchiornis, even more extraordinary, this crest and these flashes of black and white, this is for display. So independently, we suggested, and this is speculation, of course, that these color patterns and the regularity of them, and the fact that they are located on the head, wings, and tail, by comparison with modern animals, this is what you see, of course, in birds that do displays, and indeed other animals that do displays. The color patterns, the startling patterns like the black-white They're in the wings because in normal life, the wings would be folded. And then if the dinosaur is showing off for whatever reason, signaling, it would flash the wings open and you get this flash of black and white unexpectedly. That's speculation, though. So here's the reconstruction by Bob Nichols. He's showing the additional evidence of the bandit mask. And so what we claimed in our book, which I'll mention again at the end, is that we were able to show 15 dinosaurs and other prehistoric creatures reconstructed in considerable detail as life reconstructions where we could go around. And in my text, this is what I do. So Bob Nichols made this wonderful, wonderful painting, illustration using digital methods. And in the text, I went round the whole anatomy to explain this is how we know. Counter shading underneath, gingery color on top, ginger and white stripes on the tail, bandit mask over the eyes. All of this is based on evidence, based on specimens. Oh, and here's the book, Dinosaurs, New Visions, paperback in 2025, if you're interested to follow up. So I'll just show you a couple more examples just to show what's currently known, and then I'll kind of wrap up and I'll be ready, very happy to try and answer any questions. So here's a different dinosaur, Psittacosaurus, where we've got not only mummy, we've got babies. And we're showing quite a detailed pattern of color with dark color, counter shading, highlighted dark horns, speckles over the shoulders, and these weird things on the tail, which are called feathers. They don't look anything like feathers, but they're, as far as we're aware, they're made of keratin, just like hairs and feathers. How do we know about babies? There are loads of them. There are wonderful localities, Lujiatun, also in Liaoning, where you find clutches of babies buried within volcaniclastic sediments. So Lujiatun is called the Pompeii of China. And how do we know these are not hatchlings? Well, my student, Zhao Qi, cut sections through the bone, and he was able to show that all of these are 2 years old, except for the pink one, number one, which is 3 years old. So whether they're all siblings or not, we don't know, but it looks as if they might be. And this is big brother or big sister kind of running with the babies. And there is one amazing adult specimen in the Senckenberg Museum in Frankfurt, which shows these weird quill-like structures down the tail, and importantly, is covered with a very clear outline of the skin. And notice patches of dark color, paler color at the back of the calf and underneath. And this speckle, here's the speckle over the shoulders. And then here's some gut contents. So it's a plant eater. So this rather solid looking mass is semi-digested plant material in the gut as the animal died. And here is Bob Nichols's a very detailed reconstruction that was in the scientific paper by Jakob Winter, my colleague in Bristol, where they're showing multiple views, almost engineering drawing style of the Senckenberg specimen. So I flash you back. There's the specimen, the evidence of color on the skin, and this is its speckles and brown and low counter shading and a sort of yellowish gingery kind of color for these feather-like structures and the dark horns. And here in more detail then is Bob Nichols's later drawing that he did for the book. You can do it for pterosaurs too, and they're famous for also having a whiskery, feathery kind of covering. And people debate what they are. They've long been called pycnofibers. They've actually, the insulating covering of pterosaurs has been known for a very long time, and that's very uncontroversial. And in 2019, we published a paper where we suggested that perhaps these pycnofibers are also feathers. They appear to be structurally and chemically the same as the feathers of the birds and dinosaurs. And for example, what hadn't been observed before is the complex feather-like branching. So previous to our paper, people had thought that they were all just simple bristles. But we found lots of examples where they appear to show branching of different kinds in a feather. And subsequently, other authors have also reported the same phenomenon in different specimens from different parts of the world. So here's our reconstruction of this happy-looking pterosaur, all nice and fluffy, with whiskers for detecting prey, This is an insect eater, and it's just spotted lunch. And even some of the crazier pterosaurs. This is a giant one, and from other parts of the world. This is from Brazil. It's the most preposterous skeleton when you look at it. And to work out, this is the skull. Here's the eye, just to help locate you. It's got this long crest at the back of the head, this enormous sort of nasal opening, and a weird structure in front. And the fossils show us that this was spanned by skin, like the sail of a ship. And then the skeleton, the body, the wings, they just seem to be relatively small compared to this crazy, crazy head. But the head is very hollow and it's as lightweight as you can imagine. And here's the skin. And these marks are not scratch marks by a preparator using a chisel. They're actually folds in the skin. And by looking at microscopic sections of the skin under the microscope in the scanning electron microscope, Brazilian colleagues have suggested that the skin was brightly colored and that the different species of Tupandactylus may have had different bright patterns on the head membrane, and that some of them, they say, might even have been photoluminescent, like the spots of color on certain insects, glowworms, would pick up energy during the daytime, and then they can transmit that energy in the crepuscular gloom. So here is Tupandactylus, which, to just orient you, has got a 5 meter wingspan, 15, 16-foot wingspan. So this is bigger than any bird, and it's got that utterly mad-looking head. So the final example I'm going to show briefly is a very different kind of dinosaur. This is from Canada, from the Athabasca tar sands, which some of you may have seen or heard about. They are what they say they are. They're sands full of tar. Geologists are simple folk, and they call things what they are. And great trucks lift tons and tons of this gooey, tarry sand and take it back to the plant where the oil is removed from the sand and used for various purposes. And this skeleton was found a number of years ago and was named Borealopelta, which means something like northern armored beast. So this is the head at the right-hand end, and then it's got these extraordinary plates around the neck, and then the body area with a sort of metal supports towards the back, and the hindquarters are missing. And just to give you the size of it, I can't remember, it's probably 5 or 6 meters long. Here's a person looking at it. And so here is the aerial view of the whole skeleton. The head now is at the left-hand side. So it's got a unit of the back and of the neck and the torso. And there are these huge sickle-like plates over the shoulders, and then a bit of the tail area. And it's all looking a bit gingery. So what the people noted was not only are the bones of the skeleton, of the dermal skeleton preserved, because this dinosaur had bony plates within the skin, just as crocodiles do today, but also the horn covering, the keratin covering, because in life, of course, in a crocodile, the bony plates are covered with keratin. You can't have naked bone exposed to the elements. So they would have been covered in keratin, and indeed this is the case in parts of this Borealopelta skeleton. So it's reconstructed as gingery in color, quite large, and to give you the size, these scythe blades over the shoulders are 1 meter in length. So Boudica will have to look to her laurels in terms of this dinosaur. It's a humble herbivore, but it would have been preyed on by animals almost as terrifying as T-Rex. So no doubt in life this could tuck its neck and head, neck and legs and so on underneath, and would present a kind of rocky and quite dangerous exterior. And any of these sharp bony plates along the tail or over the neck and shoulders could have killed a predator who got in the way. So I just want to do a little side overview of what this all means. I've talked mainly about fossils and color, but just in a quick run through, we now apply these rules and these approaches across the whole of paleobiology to study things like speed and bite force and feeding style and growth rate. And so we do have exceptional fossils. This is one of our glories. We love the fact that we find amazing fossils. But the second assumption is critical to all of geology, which is that we apply the same standard rules of the way the earth works, the way animals work, to the life of the past. And I should have written the word on the slide here. Geologists grandly call this uniformitarianism. In other words, the assumption that uniform conditions apply. We don't assume that gravity is different in the past or that rivers work in a different way or volcanoes work in a different way. Likewise, we don't assume that dinosaur skeletons were in any way functionally different from modern skeletons. So let's just briefly look at some of these. How do we know about speed? Here's a formula that gives you the speed of any animal from its trackways, whether it's a quadruped or a biped. How do we know that it works? Because it always works with living animals, where you can actually film them and calculate the speed. And this has been done, of course, for humans and horses and greyhounds and ostriches. And therefore, this formula allows you to calculate the speed for dinosaurs. What do we need to know? Well, gravity, we know that. Stride length, you can measure from the track. Here is the right foot. Here's the next right foot. And the stride length is, say, the distance from the back of the heel to the back of the heel of the next imprint of the same foot. And hip height is worked out from the size of the print. And that gives us an idea of the size of the animal that made the track. And then we can measure that height from a skeleton. So those are the two things you need to know. And of course, it's not unexpected that as an animal runs faster, its stride length increases. So we expect that this formula will work because stride length is proportional to speed, but you must factor in the size of the animal as well. And this can also be calculated by estimating muscle volumes that are necessary. And so this was done by John Hutchinson at the Royal Veterinary College a number of years ago. He thought about what would a chicken be like if it was scaled up to the same body mass as T-Rex, in other words, the 6-ton chicken. Normally a chicken is half a kilogram, here it is, and a chicken has got good running ability, and certain small dinosaurs are in that kind of area, and so they've got good running ability. But T-Rex would have been a good runner when it was just born, but very quickly, within a year or two, it's rising up this growth curve. So this is body mass on the x-axis. It's going up the growth curve to the 6-ton chicken level, at which point running is impossible. It just wouldn't have enough muscles in its legs to shift its body. And when it comes to bite forces, we can also measure those, and we can corroborate from evidence of two mechanisms. So the first way is to look at bite marks in bones. Here is a bite mark made by T-Rex. This coin is about 1 centimeter. And by doing experiments in the lab, it's possible to work out that the bite force needed to drive a tooth 3 or 4 centimeters into the bone is something like, I think if I remember rightly, it was about 10,000 newtons, equivalent to about 1 ton of force. But that's not the maximum. That was just the force required to make this tooth mark. Other researchers, Bates and Falkingham, did virtual experiments in the computer where they take a 3D model of the T-Rex skull shown in white. They reconstruct the vectors of the main muscles shown here in red. These are the jaw muscles. The hinge of the jaw is shown in blue, and bits of food are modeled as these yellow things. And they were able to then model the forces exerted by these muscles, and they came up with forces up to 57,000 newtons, which is much, much greater than the great white shark, which has got the highest bite force of any living animal. And this, you can roughly imagine what these mean. 10,000 newtons is roughly 1 ton of force. So the great white shark can nearly exert 1 ton of force, the equivalent of a ton descending upon you with some sharp teeth underneath it, whereas T-Rex could go 6 times that. And taking this a little bit further, paleontologists use these different engineering methods. This is multi-body dynamic modeling, which is used to estimate forces and scaling that's needed to make a complex machine with moving parts. And complex structures made of different materials are investigated using finite element analysis. This is another engineering method that's used for designing bridges and buildings and aircraft and so on. And the great thing about FEA is you make a perfect 3D model of your structure in the computer. You can then test against different materials. So what if this airplane is made of 1 inch thick steel? How does it bear up? What are the forces? And the danger points are shown in red. That's where the forces, the stresses are getting very high. Then you could say, let's make it lighter. Let's make it 0.5 centimeter thick steel. How would it then work? Or what about making it from aluminium? How would it work? Would it still stand up? How about making it of green cheese? Would that work? Probably not. And so the wonderful thing is you don't risk lives. And therefore you do the same thing. This is what Emily Rayfield, my colleague in Bristol, does. She scans the T-Rex skull. You convert it into a three-dimensional wire structure like this. Each pyramid within that wire structure is called an element. And it's a finite element because it's defined very precisely within the 3D structure. You can then apply different material properties to each of these pyramids. And indeed, it's obvious that the teeth have got different material properties from the bone. The teeth might be, for example, more brittle, but harder. So they're less able to bend, but they're able to stand a great deal more compression, for example, than parts of the skull. And different parts of the skull might be made of compact bone, i.e. solid bone, or more open woven bone. And you can then model it with all those different properties all over the thing. And then you can apply different forces, give it a shove here, give it a shove here. And what are the forces that you get? And red always is the sort of danger level. Blue is quite low stress. Yellow is somewhere in between. Red is getting to kind of dangerous levels of very high stress. Anyway, so there we are. And so the stories are in the book. And if you're interested to read a little bit more about modern paleontology, we talk about it in there, giving you the kind of very detailed evidence of all this very recent work. And this one focuses very much on color. And I've written other books that talk about other stuff, but I'll just pause at that point and stop sharing. So thank you very much for your attention.
Thank you very much, Mike. That was fascinating. Now then, does anyone have any questions? I'll just check what's in the chat and see if we've got anything to relay to you from there. Okay.
Yes, I can go through the chat
Oh,
ones if you
thank
like.
you.
And I'll read them out. So, yes, a very good point from Paul. At times there was more oxygen in the atmosphere now. And that is absolutely right. Yes, I was being, in a sense, dismissive, of course. We don't vary gravity unless we have some evidence, but we do vary oxygen and carbon dioxide because we know
Mm,
at different points, for example, in the middle of the Cretaceous, oxygen levels were quite high. And that would affect the capability of flight of organisms to a modest extent. So people do factor that in. That's absolutely right. Maximum size capable of flight. So Paul also says, what's the maximum size? I think if we had not known the fossil record, we would have said, of course, oh, well, it must be the condor or whatever is the biggest living flying bird. And I can't remember the wingspan. Is it 2.5 meters, 3 meters, 8 feet, something like that?
huge.
Whereas when you look at fossil birds, there are one or two even bigger ones that maybe are half again as big as the biggest living bird, flying bird. And then when you look at pterosaurs, some of those flying reptiles, they got to be even bigger. Some of them maybe had a wingspan of 13 meters. So whatever that is, that's about 40 feet. That is absolutely crazy. So double the size of the biggest winged bird. And yet all the evidence suggests that those pterosaurs, they had fully developed wings, that the wings were in proportion to their estimated body masses, and that therefore they must have been capable of flight. Although it's very hard to comprehend. People have tried to make models, but it's actually very difficult to make a physical flapping model. And I think it's much easier to do some of these experiments. Engineers find it easier to do some of these experiments in the computer,
Yeah.
where you build your 3D model in the computer and then you can test it in various ways. Yes, so let's see. Yes, so Paul also follows on the surface solidity and composition also affect the speed, e.g., wet mud, dry soil, tall grass, rocks and trees, bushes to dodge around. Yes, I think this is about animal footprints. So people only look at cases, I think, where the animal is moving at a steady pace. And of course, generally, it's true that what you see in the fossil footprints probably isn't the maximum speed.
Mhm.
Because to preserve the footprints, there has to be a certain amount of water in the sediment. If it's just purely dry, like running over sand dunes, very dry sand dunes, the footprints quickly disappear. And so probably the speeds that we're getting for dinosaurs are not their maximum. But nonetheless, I think they're fairly indicative. So at least it put an end to those futile debates that were going on when I was young. Oh, T-Rex could run as fast as a racehorse, relatively speaking. Say, no. And I think engineers would have said, no, don't be ridiculous. It would break its legs. It just couldn't do it because there are safety factors and the dimensions of the bones have to be sufficient. And essentially, as you run faster, you are effectively increasing your body mass more or less, so that the impact of your foot on the ground, as I'm sure you know when you're running for the bus, you think, oh, for goodness sake, I shouldn't be doing this because you're hurting your knees because you're expressing 2 or 3 times your normal body mass as you run. Um, Dave Fleet says the past 10 to 20 years I've heard described as a golden age of dinosaur research. Yes, 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 absolutely. What advances do you see coming down the track in the next years? Oh, blimey. Well, I suppose there'll be more of the same because, of course, I've tried to cover, I didn't talk about growth and babies and eggs. And so I suppose you think about the different biological functions. There's obviously feeding, there's locomotion, there's growth, there's sex, there's display, there's all sorts of other social things. There's also senses, I suppose you could say. How well could they see? How well could they hear? Balance, I didn't talk about that. People can scan in quite a lot of detail the structure of the brain of a dinosaur in certain cases, even the cranial nerves. The semicircular canals can be scanned in a lot of detail. And they can actually tell you something about the pose of the head, because I'm sure you're thinking back to your school biology, the semicircular canals. You've got a horizontal semicircular canal, and the clue is in the name. It's got to be kept horizontal, or you don't feel right. And that's when you get travel sickness, when you're bouncing around. And so, of course, oh yeah, if you look at a dinosaur, you can detect, because they're set into the bone, we can look for that horizontal level within the bone of that semicircular canal. And more or less that lines up and tells you the posture of the head when it's at rest. Is it up or level or partly down? The sort of angle at repose. Um, David Winsford asks, might feathers have evolved to improve the gait for speed? That has been suggested. Yes, because some of these feathered dinosaurs they had feathers on the hind legs, which when they're running along would look like cowboys' chaps. They look like cowboys' feathered trousers. And I'm not sure whether running in leather trousers with feathers out at the back would make you run faster. Probably not. But then again, they wouldn't be suffering problems of their trousers falling down. So I suppose if the feathers are actually cemented to your leg, I'm not quite sure. I think they would slow you down actually, because you'd think, oh, what do you call it? Not air, improve your aerodynamics. But the trouble is your legs have to move forward, and so they're moving into the wind, and so you'd be carrying all these feathers, and so I'm not quite sure. If you were on a row, if you were standing static on a roller skate, on a skateboard, yeah, yeah, they might improve your aerodynamics by diminishing eddies behind, which would slow you down. But I think for running, and I don't think they had skateboards, so I think those feathers are for display. And even if they're an impediment, think of the peacock, the male peacock tail. It's utterly stupid. And it's a real impediment. But nonetheless, this is what Darwin's insight. The tail of the peacock is an impediment in terms of natural selection because these peacocks are making themselves very visible and slowing themselves down because they have to carry that blasted thing around with them. So they're much more likely to be eaten by predators. But the benefits of sexual selection are much better. You get many more wives, therefore more babies, therefore that's all that matters in terms of evolution, I suppose. Oh, Martin Itaker asks, how many staff working in Bristol Department of Vertebrate Paleontology? We're a paleontology group as a whole, so colleagues are working on in marine fossils, invertebrates, plants, all sorts of different things. There's about 10 or 12 staff here across the board studying everything from genomics of evolution through to plankton and the evolution of climates. There's a broad sweep of important areas of impact of paleontology. It's not all dinosaurs. Dinosaurs, for me, are the fun bit, but they're entirely useless. Whereas the study of plankton can allow you to reconstruct climates in a lot of detail, for example, which is quite important. Colin from Portsmouth, is it possible to distinguish between gliding flight and powered flapping flight? It's quite difficult. I mean, clearly in the modern forms, yes, of course, but it's quite difficult from a fossil to be sure, because gliders, of course, in certain cases where the wings are insufficient to hold the body entirely up in the air, then if there are body extensions of some kind, like in, I don't know, flying squirrels, or some of you may have seen those amazing pictures of flying frogs. You'd think how utterly ridiculous. They've got big feet with skin between the toes, and they can fly by spreading out their big feet. Utterly bonkers. And clearly they're not flapping. They're just jumping. These are tree frogs. They jump from tree to tree, and presumably by spreading out their feet, they can slow down the rate of descent. Yeah, so that's one way of telling the difference. So there are quite a lot of definite gliders in the fossil record, not birds or dinosaurs, but lizards and mammals and so on, like the modern ones, that were capable. So I think people in aerodynamics use the term flight for any kind of movement through the air, including an ash key falling from the tree. It is flying because it is sustaining a kind of movement. It's not simply falling to the ground. So it's slowing its descent by the nature of the wing-like structure. And so any animal that's capable of slowing its descent is called a flyer. But powered flight or flapping flight is the thing. Yeah, so it's difficult to know, and I don't think that frog is ever going to become a flapping flyer. That would be a crazy kind of evolution. But clearly there were multiple efforts among these early bird-like dinosaurs at flight. And at the time, you couldn't have said which of these, Microraptor with its four wings, it could have prevailed. And all birds today might have four wings for all we know. But for whatever reasons, the Microraptor solution to flight was good in its day. It enabled that animal to jump around in trees and fly from tree to tree to get its prey or whatever it was doing. And likewise, Chi, that dinosaur with membrane, bat-like membrane as well as feathers, it in its own way was flying, but didn't give rise to anything else. Uh, yes, so Diane Newell, what does the evolution of color and feathers tell us about vision and color? Yes, so of course, as soon as you see that they have patterns, that shows there's a certain visual acuity. Probably the colors would indicate or suggest that they may have had some form of color vision. And that's something we can't say for sure. I think all birds do. I'm not sure about crocodiles, whether they have color vision. Because when we're looking at the unknown in fossils, we can look at the living relatives and we think in terms of a bracket. So dinosaurs are entirely extinct, but they're bracketed by birds and crocodiles. So birds and crocodiles share a lot of features, but I'm not quite, and therefore, if they share a feature, we can then infer that probably dinosaurs had that same feature as well. That's why we can't say that dinosaurs sang beautiful songs, because although birds maybe do that, crocodiles definitely do not. And therefore, we can rule out that bird that probably dinosaurs did not sing beautifully like certain modern birds. But on the other hand, the muscles of the leg of a bird are the same as the muscles of the leg of a crocodile. Therefore, we can probably say the muscles of the leg of a dinosaur are probably the same. But no, we'd love to know about vision. Malcolm Greaves asks, is it possible to state why the Chinese sites has generated so many perfect fossilized? Yes, I can't. And it is a great question. And people struggled for decades to dig out 15 specimens of Archaeopteryx in Germany. And these Solnhofen beds in Germany preserve beautiful, beautiful fossils, soft tissues, things like worms and jellyfish and all kinds of stuff. Why are we not finding thousands of birds? This is certainly a big incentive. Those quarrymen in Germany are not failing to find them because they can't be bothered. If they find an Archaeopteryx, they get a huge amount of money. They are going to look for them, but they're just not there in such numbers, whereas in China they are. And I think partly it's because these fossiliferous beds extend over a very large area. I think the ones in Germany maybe just cover a few acres, whereas these ones in China they span over thousands of kilometers. They're just much of the north of China it has these kinds of sites. And the way they're preserved is they were deposited, they're in lakes, and the sediment comes from volcanoes. So it's sort of acidic. And it's not directly from volcanoes, except at that one locality, Tun, where the hot ash, was falling on the dinosaurs and encapsulating them in the ash. Mostly it's ash that is being reworked, as we say, picked up by the rain and the water and washed into the lakes. But probably the slight acid in the water, like in peat bogs, preserving the peat bodies. Peat pickles skin. And so these amazing bog bodies of humans that people dig up, they are preserved maybe in a similar kind of way. But who knows? It's amazing. And it did seem as if people were finding them to order. People would sort of say, I remember when the first things were being announced, oh, what if we could find this? Or it'd be wonderful if we could find that. And blow me, somebody does. Um, Philip Dan says the sail on the head of that strange looking dinosaur was made of foldable skin. Was it the forerunner of the cockatoo for display? No, this was a pterosaur. So the pterosaurs are cousins of the dinosaurs. So they split apart in the Triassic. So the pterosaurs, the flying reptiles, are quite distinct from birds, and they evolved flight independently. They may have acquired feathers together. It may be that feathers occurred right at the start of both. But they split apart maybe 240 million years ago, and the pterosaurs went off and did their thing, and the dinosaurs went off and did their thing. And they gave rise to birds at maybe 160 or 170 million years ago. So that head structure is entirely weird and unique to pterosaurs. And we know some birds have got head structures like the cockatoo, but they're nothing like as mad as the pterosaur ones. Colin from Portsmouth again, you can't see muscle attachments in the bones that would be absent from gliders. Well, you can. So the pennaceous feathers, I think the whiskery feathers, the down type bristles and so on, do not embed into the bone. They are simply within follicles in the skin. But the pennaceous feathers, the ones with a quill, they do. And so along the ulna, which is the bone along the front of your arm, and the humerus, which is the upper arm bone, in a lot of these feathered dinosaurs and birds, you can actually see very regular pits in the bone. So when you get a chicken, if you have a look at the two main bones in the wing, you may well find those pits where the feathers would implant. Although I think that would be a measure that you've got an organic bird, because most of the ones we eat are far too young, and so the feathers are there, but they're really not. The bones are not fully ossified, and it would just be in an older one that had been stomping about a bit you might see that. But nonetheless, yes, there's regular pitting. And so even if you don't find the feathers, you can tell from the skeleton, oh, this thing had pennaceous feathers. And you could even count the numbers. You can say, oh, yes, there would have been about 7 or 8 along the forewing and another 10 along the hind wing or something like that. Feathers good for insulation or hairs better? I don't know. I think it depends on how they're organized. I think feathers are better in a simple sense because one follicle can produce multiple branches. So obviously a whiskery feather branching into lots of strands immediately makes it better than a single hair. And mammals are incapable, or our hairs don't branch. It's just a definition of a hair versus a feather. Although developmentally and in terms of evolution, mammal hairs and bird feathers have a deep shared origin, way, way back in terms of the fundamental genetic control. And they're made, they come out of follicles, they're made of keratin, they share melanosomes and melanin color, blah, 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 blah. Um, so yeah, but then again, I don't I forget the detail of how the feather how the how the hairs of a polar bear work. I think they have certain kinds of hollow. Well, that's another thing actually. Feathers are hollow, and hairs are solid usually. But then I think polar bears, if I remember, they've got some specially adapted hairs that may be hollow, and then that begins to increase the insulating properties. And oh yes, so and Colin, the large muscles that flap the wings, yeah, yes. So although the muscles are not themselves preserved, because muscles tend to break down very quickly after an animal dies, there are markings of where the feathers were. There are markings on the bones of where the main muscles attach, because muscles are fixed onto the bone with special fibers called Sharpie's fibers, and those fibers go right into the structure of the bone and they align with the direction of the muscles. So that if you look at cross sections of the bone of a fossil animal under the microscope, you can, and if it's well enough preserved, you can sometimes see these fiber directions actually within the bone. And those mark the orientation of the muscle. Well, they tell you, first of all, there was a muscle there. And if you look at over the area, you can work out how big it was because the cross section of the muscle will show up on the bone. And the cross section gives you a measure of how strong the muscle is, and also the orientation from that. So, yes, it's sort of painstaking work, but you can work out where the muscles are. And the handy thing is that probably more or less 90% of the muscles in a T-Rex are the same as what you've got. So it's not a total reworking of the evolutionary palette, because they work pretty much. All tetrapods share pretty much the same muscles to operate their arms and legs. And these are taken over and enlarged for flight. So, of course, when you eat your chicken, the biggest muscle is the pectoralis. That's the one that pulls the wing down. And that's the muscle that bodybuilders try to develop by using chest expanders. And then the other bit of muscle, when you pull the breast meat apart on a chicken, there's a smaller bit. That's the supracoracoideus, which pulls the muscle, which pulls the arm up. So there's a pulley system over the shoulder in a bird that means that the supracoracoideus contracts, but it actually pulls the wing up. And then the pectoralis contracts and pulls it down. And the pectoralis is about 10 times the size of the supracoracoideus because it has to be for flight. But we have both those muscles, although we don't fly. Oh, I bet so. People got hands up. Oh, this is the last one on the chat. So Pete C from Swindon, fossil formation fascinates me. Can you give examples where we think this process might be taking place today? Will there be fossils of hedgehogs and magpies? Probably. So there's not fossilization taking place everywhere. In a nutshell, we can only really get fossils where there is sediment accumulating. So typically the bottom of lakes and the bottom of the sea, shallow waters, but not too shallow, because inshore water is stirred up by the waves and constantly churned and dumped and then rechurned. So generally within the wave zone, down to deep water wave base, the sort of storm water wave base, you're not going to find much preserved. So typically we find fossils in that shallow sea area, maybe deeper than 50, 60 meters offshore, and often in lakes, because lakes are often quite gentle sites of accumulation of mud and sediment, and anything that falls in can often be entombed. Hedgehogs and magpies typically die in woods, and that's not a great place for fossilization because soil is being churned by earthworms that will happily eat their way through any carcasses, and the soil is sort of being reworked by rainfall and falling leaves and all sorts of other stuff. So, yeah, if you want to be preserved, lie down in the middle of a lake or get a rowing boat and go offshore maybe a mile or so and then just jump in. So Neil Allen has got his hand up.
Yes, you mentioned one time about scanning tunneling microscopes, but on and is some of the color being discovered by photonic crystallized, nanoscale photonic crystallization color? So you're actually looking for the nanoparticle sizing or anything? Is that the way that you're doing some of the
So
color?
it's not that small, although it is
Oh,
pretty
okay.
small. So the size, I should have said, the size of the melanosomes is 1 micron,
All right. Okay.
which is the limit, which is about the limit for a normal kind of SEM. So we're not using any fancy kind of imaging device. And you're right, of course, there are many that would take you to much higher levels of detail. And so there is more to be done, I think. We've done a little bit of work on what's it called, FIBS or TIBS, I can't remember, the kind of electron microscope where you can actually cut across and observe and that kind of thing. But we can do it on a regular SEM, which is nice. So even our students are doing this. They have a beat up old 20-year-old scanning electron microscope that they can use, and it's easy enough. So Barry Zussman, you have your hand up.
Yeah, thanks, Michael. Great talk. My question is also related to melanosomes. You mentioned there are these two types, one sort of greys and black, grey, white, and the other is sort of ginger. So I can see how those can create the greys and the browns.
Yeah.
What about all the other multitude of colours we see
I'm
in
glad you
birds?
asked.
A lot of them are actually done through light refraction rather than
You're
pigment.
right. You're right. So
Where do all the others come from historically?
yes. So birds are much better at color than mammals. Mammals generally we're limited to those melanin-based colors, and that's that. Whereas in birds, you're right. They've got, first of all, there are other colors that are non-melanin that are produced by food proteins. So carotenes produce yellows and reds. So famously, of course, the color of the flamingo. If it's not getting the correct diet, it looks a bit pale. And it's not sickly, it's just that it lacks that red color that's coming from algae within certain krill-like things that it's eating. And the other food-based colors are porphyrins, which are also proteins, and they give purples and greens. And so we don't know whether dinosaurs would have also had some of those food-based colors because they are not encapsulated within any structures. They're just within the feather, and they don't leave a trace. So far, people have been looking at the chemistry of melanins, and melanin is very recalcitrant. It survives remarkably well against high pressures and high temperatures of burial, but it does modify. And so there's still a debate about how much of what we're seeing in the fossils is real keratin or real melanin. They are very tough. They do survive a long time, but they also probably are modified. So we need to be quite careful with chemical mechanisms because you're probably not going to detect the modern molecule. It'll be some modification, and then you get into complexities. So we're happy that we could do this on an ultra-structural basis, which is a little bit less dodgy, if you like. And experiments have shown that melanosomes shrink. They lose fluid, but they don't change shape. So if they start spherical, they remain spherical. If they start sausage shaped, they remain sausage shaped. So, but there's still a lot. That's another area. Somebody asked a question earlier about what next. There's a lot of interest, of course, in molecules, and people did make mistakes in the 1990s when they were claiming that they had got DNA from dinosaurs and we could bring dinosaurs back to life. Any chemists amongst you will know that DNA, the nucleic acids are at the other end of the chemical scale of hardiness. They're very labile. They break down within years, if decades. Millions of years, no chance. They're just gone. And so that ranking of organic molecules was in our favor. Melanin is tough, and therefore it probably helps the preservation of the hairs and feathers.
Hmm. I have a question for you as well, Mike.
Yeah, thank you, Mary.
Just looking back, I've heard a view that say 1 in a million gets preserved as a fossil, and you've obviously got, particular circumstances that you need for the animal to be preserved. And from what I understand, it's mainly shallow waters, as you've described, or deserts that have been the environments that we've found most of the fossils in. Does this leave us with gaps in the fossil record in terms of understanding how the environments all worked?
Yes, so the fossil record is notorious for the big holes in it. That's absolutely right, because fossils typically are preserved only in certain circumstances. Of course, I was talking a moment ago about those cases of exceptionally good preservation, but there are fossils known from fossil soils and from riverbeds and from other kinds of locations, and indeed from deserts. There are some amazing mummified dinosaurs that have been preserved in deserts because, of course, by drying out quickly in certain circumstances, the whole carcass can be more or less preserved. And there are these mummified dinosaurs where the skeleton is there, and then there's a strange kind of bag around the skeleton, which at first people would chip away. What's this rubbish? Get rid of it. And then they realized they were hacking through the skin, so the whole skin structure around the dinosaur was actually there. Although how much chemically of the original skin actually was there is a debated point. It was probably largely an impression, but a very good quality one. So you could see the individual skin cells and the structures of the skin and such like. Yeah, and then the gappiness, yes, it's something we're aware of. I'm careful to talk about cases where we have exceptionally good fossils, where we can really go to town. And you wouldn't attempt to do this kind of work on many, many types of fossil locations because the data just aren't there. The fossils are not good enough.
So I guess on that basis, we'll never really know how many species there are. We can look at the plethora we have today, and the extrapolation would be similar numbers in the past.
You're absolutely right. So it's actually difficult enough to count the number of species on Earth today. You can look up Wikipedia and you'll find accounts of thousands and thousands of species. You can look at the entire scientific literature, and I think you'll discover maybe 1.5 million species have been named of living things. But people still estimate there must be at least 10 million species on Earth because, and the evidence for that is that when coleopterists go to remote parts of the world, they find new beetles at the rate of about one every 10 minutes. So the only limit on our knowledge of beetles is the amount of time that coleopterists have got to look at them and write the papers giving them names. Whereas probably we've seen and identified most of the living birds and most of the living mammals. And when it comes to deep sea creatures and certain kinds of plants and microscopic organisms, heaven knows. So our knowledge of living species total diversity is quite limited. And it's the same with the fossils. But I think the approach people would use would be a sort of sampling approach. So the way we know that there are probably 10 million species out there, even though we don't have a catalogue of them all, is that people have done intensive studies of specific parts of the world, like a particular coral reef or a particular tropical rainforest, and they look at everything in really intensive way with dozens and dozens of colleagues. And so after a year or two, they can say, okay, we've now collected everything from this 1 hectare patch, and this is what we find. And then you can scale that, of course, to the wider world. And that's where the 10 million figure comes from. And I
Your
think
comment
also,
about, sorry, your comment about the beetles reminds me of Darwin's comment about God must have loved beetles.
yes, that's why I chose them. That's right. I know. And the trouble is there just aren't enough coleopterists in the world. And it comes down to pounds, shillings, and pence, because obviously there are probably 3 or 4 in the Natural History Museum who don't sleep. They work day and night because there's no limit to the numbers of new species. But they would probably say, oh no, we need 100, immediately. The government should be immediately investing. We need 100 coleopterists right now to get on with the work. But they're not going to.
But I mean,
Yeah.
the wonderful thing as well about the creatures that sort of have shorter lives is they are evolution in action. You can actually, over sort of several human lifetimes, perhaps see evolution happening. Peppered moth obviously is one.
Yes. Yeah, that's right. So when you're looking at fossils, you have to be aware of that. That's right. Certain species turn over very quickly, so that you might find 1,000 specimens of a particular species of fossil beetle, but only 3 specimens of a bird
Mm.
in the same locality. But you're quite right. You'd have to then take into account. It would be wrong to say for every bird there are 500 beetles,
Mm.
because the beetles might represent, in both cases, you might be looking at a 10-year sample, and the beetles have just been turning over and dying and dying and dying, and the birds have lived a bit longer. We call it time averaging, so paleontologists are very careful to pay attention to that.
But all the new techniques that we have now must be wonderful in terms of trying to extrapolate backwards. Because yes, we have to look at what's alive now and extrapolate back in terms of what previous animals would have been like and the faculties they would have had. And I'm
Yeah,
thinking as well in terms of things like birds being able to see different wavelengths to us as well. Because I understand that sort of as primates we lost colour vision and then got it back again. So we're
yes,
quite
yes,
odd in that regard.
yes.
We don't know what we lost.
And I think that's right. I think you can predict a lot about the past from living organisms alone.
Mm.
But if you even add a very small number of fossils, it immediately enriches the knowledge hugely. Because looking at modern organisms, you would never predict the existence of dinosaurs.
No.
The further back you go, the less certain you are. I mean, Darwin got it right in the sense that you can look at a virus and a daffodil and a human, and you can show, of course, that they are all related. They must have had a common ancestor at one point in the deep past. To work out how long ago that common ancestor was, you do need to insert a few fossils here and there in your evolutionary tree to give it some dating. It doesn't change the pattern of relationships. The virus is here, the daffodil is here, the human is here. And so Darwin was the first to really strongly say all living things
Mm.
are related back to some ancestor. And the other point, of course, is you would never predict trilobites or ammonites or dinosaurs from the living organisms. So the further back in time you go, the less certain you are.
Mm.
So that's another exciting area of paleontology that I haven't talked about, which is that lots of our students are working closely with people who are studying modern forms and trying to understand how particular groups diversified. And we can tell, for example, beetles are hugely diverse because they diversified hand in hand with the diversification of flowering plants, which really got going in the Cretaceous and led to an enormous boom of life on land, and particularly in the tropics. So we can enrich our understanding of modern biodiversity, biogeography, by looking at the dating of what was going on and some of the fossils. And of course, for fossil beetles and insects, there are fantastic specimens in amber in the Cretaceous, which just would blow your mind away. And the detail is just astonishing. So yeah. And the other area I didn't talk about that's really important is climate change. And of course, we can only speculate about climate change over relatively short time spans if we use historical records like tree rings and ice records. But if we expand to include the fossil record, then of course we can go back and look at much longer time scales. And I think it's important to know that the dating is getting so much better. That's another area that has really advanced enormously in the last 30 years. So one of my other interests is the end Permian mass extinction, which happened 252 million years ago. And when I started looking at this in the 1990s, people were very woolly about actually how big was it? Did it all happen at one time? What's the date of it? Many people said it maybe lasted over 50 million years. It was a long span of turbulence and perturbation. Whereas now we can say there were two pulses of extinction separated by 60,000 years.
Mm.
So the quality of dating has really, it's warm. I wave my hands about and it puts my hand up. So, anyway, I'll stop waving my hands about.
No problem. You're mentioning about the evolution of flowers and beetles. It does make me wonder if that contributed to birds' vision and the colours they can see and perceive. Because obviously insects have the ability to see UV as well, and they're
Yeah,
pollinators to a
yeah,
degree the same as some
yeah,
birds.
that's right.
Just thinking about, the patterns on sunflowers that we can't see, but they
Yeah.
can.
I think you're right. And I think a lot of the color range, and I didn't mention, somebody did ask, I think Barry asked about colors. And yes, there are iridescent colors in birds, and people have claimed to have found them in fossils as well. And a lot of the iridescence comes from alignment of the melanosomes, so that if they're arranged in sheets, all pointing the same way, of course, then you get a nanostructural effect like in a beetle. And of course, the carapace of a beetle is particularly these jewel-like, amazingly brightly colored ones. The carapace or the wing covers, the elytra, are maybe they're a 10-layer nanostructure where each layer is semi-transparent, and so the incident light is partly reflected and partly passes through each of those 10 layers. There may be a base layer in the chitin making up the elytrum, which is a bright color like green or blue or red. And then the 10 nano layers over it are partially transparent, and they massively enhance that color and give it that iridescent jewel-like property. And yes, you're right. Of course, birds and many insects and birds can see over a wider range, including some ultraviolet and infrared. So they see the visual range that we see, but they also can add some UV and some IR. And therefore, that's right. There's amazing photographs of flowers showing their ultraviolet patterns that we just don't see.
Mm.
And yet they're providing a sort of landing by. And it's fascinating.
It is. It is. Right, we don't seem to have any questions in the chat. Do we have any more questions from people to pass through to you?
Yes, Mike.
Mike, hello. Thank you for your things going up and down. That's very kind.
I'll give you a question from the skeptical point of view to the dino bird thing. In Sinosauropteryx, your first image was first discovered. People immediately jumped to the idea that they were proto-feathers. They've been proto-feathers, they've been integumentary structures, they've been bristles, they've been dino fuzz. I've never seen anything that looks feather-like in Sinosauropteryx. And John Ostrom, who wrote Compsognathus, couldn't see any feather-like structures in Compsognathus as well. What's your comment?
Okay, so yes, so early on there was quite reasonably a caution about being sure what these were. And they were called in the original papers proto-feathers. And they could have been called whiskers or bristles, or people could have tried to use some sort of neutral term. In the Chinese language, the word Mao, as in Mao Zedong, is a feather or a hair. So they have a word, their typical word they use, Mao, could be hair or feather, or any whisker. So it's just the curiosity of the English language, or probably most European languages, that we have to be explicit. Whereas in others, they would use a general term. Yeah, those days are past. All of these other thousands of specimens from China with complex pennaceous feathers, the preservation of the pennaceous feathers is the same as the preservation of these less certain whiskers, bristles, protofeathers, whatever you want to call them. And so the most parsimonious interpretation would be to say that's what they are. You could still maintain that they're not, but then we were impressed by the fact that they contain melanosomes. So they contain melanosomes just like hairs and feathers. So that excludes that they could be muscles or skin. Well, they could be skin, but my goodness me, you've got to bend over backwards to make a very complicated argument to explain somehow how skin is shredded into these very thin fiber-like structures. Skin, of course, can contain melanosomes, but muscle and other structures do not. So the fact that they're stuffed full of melanosomes, just like complex pennaceous feathers, I think made it multiply clear for us anyway.
Going up to your one of the last images you had of the pycnophytes.
Yeah.
Pycnofibers.
Yeah.
Just like integrating collagen fibers. Professor Lingham's.
You're breaking up a bit. I think so. Something about the pycnofibers. So
they look pictures that you showed. They looked just like shredded collagen fibers, shredded stiffening fibers.
Yeah, well, you can think that if you like. They're full of
feathers.
melanosomes, and so collagen fibers are not full of melanosomes. They occur just on the exterior surface of the creature. And I think they don't show the sort of barring pattern that you might expect with collagen. And in terms of hardiness, keratin is tougher and melanin is tougher. So, when we're interpreting fossils, you can have a number of different views, but I tend to follow the majority view and the most parsimonious view, which the most parsimonious view follows the simplest explanation, which is the usual principle in science. And you can go on maintaining there is a certain doubt, and you're quite right. Of course, there's always going to be a certain doubt with fossils. But I think I'd circle back to the beginning, which is where I explained the chain of inference idea. I think, yeah, let's go for it. Let's have a look, and you or somebody else can repeat the observations and see what you find. And if you can find that the pycnofibers or bristles or other structures don't have melanosomes, that somehow we're mistaken in that observation, then you're in business, or you're beginning to be in business. And just saying I don't like it is fair enough. You can say that, but it's not a scientific argument, I'm afraid.
The melanosomes scattered over the general area, or just concentrated?
They're just within the keratin structure. So I showed you the photograph, and so they're not scattered more widely. If it was a bacterial film, if these were bacteria, people have looked at that time and again, and they occur just within the feather structure. They're organically part of it. That was a piece of preserved keratin where you see the moldic structures and a number of spherical structures sitting there, and people have seen this in samples from many different deposits of Jurassic, Cretaceous, and Cenozoic age. And they're just the same in Neogene fossils, Mesozoic, they all look the same. So, it's QED, I think, parsimony.
I think the fact that you've seen it across different
Mike and
animals.
I have a love relationship.
I think the fact that you can see these things across multiple different creatures, different time periods, different environments, just tend to support that they are there personally, and I'm no expert. Right, any more questions? I'm sure there are loads, and some people are just too shy to ask them, to be honest. But on that basis, if there are no further questions, I'd just like to say thank you very much, Mike, for your time and for your expertise. We really appreciate it. It's been fantastic talk, and I would love to invite you back again at some point in the future if you're willing.
Very happy to. And I can
Thank
talk
you.
about
So,
something different if you like.
oh, of course. Well, I mean, as you can see, we have a ready audience for any topic.
Fantastic. Yeah, no, really pleased. Thank you so much.
Right, so can I ask you all to unmute, please, and to show your appreciation in the usual U3A way? Thank you, everybody, and thank you again, Mike. Thank you very much.
Yeah, thank you. Thanks very
Thank you
much.
all.
discussion with our
Excellent.
panel where we
Thank you.
discussed the questions which we
Okay then.
Excellent talk.
Okay then. Thank you very much, all.
All right.
I'll
Bye-bye.
hopefully
Thanks,
Bye.
Michael.
Thank you.
goodbye. I'll see you all next month, hopefully. Bye for now.